Hi everyone. In this video I am going to explain you about protoplasm, cytoplasm, endomembrane system. Under this, endoplasmic reticulum, Golgi complex, lysosomes and vacuoles. Protoplasm. Protoplasm. The liquidous living matter in cell called protoplasm. This was first observed by the scientists Corti and Fontana. This liquidous living matter, first named as sarcode by Desjardins, then this sarcode renamed as protoplasm by Purkinje. Protoplasm divided into two types: cytoplasm, nucleoplasm. The liquidous living matter other than nucleus, which is present outside nucleus, called cytoplasm. The liquidous living matter present in the nucleus called nucleoplasm. Cytoplasm plus nucleoplasm is equals to protoplasm. Protoplasm is liquidous living matter. The name protoplasm given by Purkinje. Protoplasm divided into two parts: cytoplasm, the liquidous matter outside the nucleus; nucleoplasm, the liquidous matter in the nucleus. Cytoplasm plus nucleoplasm is equals to protoplasm. Cytoplasm. Cytoplasm. The term cytoplasm was given by the scientist Strasburger. Cytoplasm is the part of cell present between the nucleus and cell membrane. Cytoplasm can be divided into two parts: cytosol and trophoplasm. Cytosol. The liquid matrix of cytoplasm except cell organelles called cytoplasm. The part of cytoplasm containing organelles called trophoplasm. Trophoplasm is also called deutoplasm. Cytoplasm is the term given by Strasburger. Cytoplasm present between nuclear membrane and cell membrane. Cytoplasm divided into cytosol and trophoplasm. Cytoplasm without organelles called cytosol. Cytoplasm with organelles and non-living inclusions called trophoplasm or deutoplasm. Next, endomembrane system. Endomembrane system. While each of the membranous organelles is distinct in terms of its structure and function, many of these are considered together as an endomembrane system because their functions are coordinated. The endomembrane system include endoplasmic reticulum, ER, Golgi complex, lysosomes and vacuoles. Since the functions of mitochondria, chloroplast and peroxisomes are not coordinated with the above components, these are not considered as a part of endomembrane system. Endomembrane system includes endoplasmic reticulum, Golgi complex, lysosomes and vacuoles because all these functions are coordinated. Endoplasmic reticulum. Endoplasmic reticulum. Electron microscopic studies of eukaryotic cells reveals the presence of a network of reticulum of tiny tubular structures scattered in the cytoplasm that is called endoplasmic reticulum or ER. Components of endoplasmic reticulum. Endoplasmic reticulum composed with three major compounds which are cisternae, vesicles, tubules. Very first one, cisternae. These are long, flattened and unbranched units arranged in stalks. Vesicles. These are vowel membrane bound structures present in cytoplasm. Tubules. These are irregular, often branched tubes bounded by membrane. Tubules may free or associated with cisternae. Remember, endoplasmic reticulum are network of reticulum of tiny tubular structures scattered in cytoplasm. Endoplasmic reticulum composed of three major components such as number 1 cisternae, number 2 vesicles, number 3 tubules. Structure of endoplasmic reticulum is like the Golgi body, but in endoplasmic reticulum, cisternae, vesicles and tubules are isolated in cytoplasm and those do not form complex. Golgi body is a localized cell organelle while endoplasmic reticulum is widespread in cytoplasm. Endoplasmic reticulum is often termed as the system of membranes. The endoplasmic reticulum divide the intercellular space into two distinct compartments, which means luminal inside endoplasmic reticulum and extraluminal cytoplasmic endoplasmic reticulum. Remember, endoplasmic reticulum is like Golgi body, but endoplasmic reticulum, cisternae, vesicles and tubules are isolated in cytoplasm, but not in the form of complex. Golgi body is localized cell organelle, but endoplasmic reticulum is widespread in cytoplasm. Endoplasmic reticulum, also called system of membranes. Endoplasmic reticulum divides intracellular space into two distinct compartments, luminal compartment and extraluminal compartment. Endoplasmic reticulum broadly classified into two types. Number 1, rough endoplasmic reticulum or granular endoplasmic reticulum. Number 2, smooth endoplasmic reticulum or agranular endoplasmic reticulum. Rough endoplasmic reticulum, which contains ATS ribosomes. These ATS ribosomes binds by their larger subunit with the help of two glycoproteins called riboforin-1 and riboforin-2 on the surface of rough endoplasmic reticulum. In smooth endoplasmic reticulum, ribosomes and riboforins are absent. Rough endoplasmic reticulum mainly composed of cisternae, but smooth endoplasmic reticulum mainly composed of tubules. Rough endoplasmic reticulum abundantly occurs in cells which are actively engaged in protein synthesis and secretion. Example, liver, pancreas, goblet cells. Coming to smooth endoplasmic reticulum, abundantly occurs in cells concerned with glycogen and lipid metabolism. In animal cells, lipid-like steroidal hormones are synthesized in smooth endoplasmic reticulum. Example, adipose tissue, interstitial cells, muscles, glycogen storing liver cells, and adrenal cortex. Let's see the diagrammatic representation of endoplasmic reticulum. See how endoplasmic reticulum associated with nuclear membrane. How smooth endoplasmic reticulum are present, how rough endoplasmic reticulum are present. Let's see this diagram carefully for your better understanding. Modifications of endoplasmic reticulum. Endoplasmic reticulum modified into two types. Number 1, sarcoplasmic reticulum SR. Number 2, microsomes. Sarcoplasmic reticulum. This smooth endoplasmic reticulum occurs in skeletal and cardiac muscles. Sarcoplasmic reticulum stores calcium 2 plus and energy rich compounds required for muscle contractions. The smooth endoplasmic reticulum, which is present in skeletal and cardiac muscles, called sarcoplasmic reticulum, which stores calcium 2 plus and energy rich compounds for muscle contractions. Microsomes. Microsomes are the pieces of endoplasmic reticulum with associated ribosomal particles. These can be obtained by fragmentation and high speed centrifugation of cell. They do not exist as such in the living cells. Scientists used microsome for in vitro protein synthesis only. The pieces of endoplasmic reticulum with ribosome particles called microsomes, which are widely used by scientists for protein synthesis in vitro conditions. Functions of endoplasmic reticulum. The very first one, mechanical support. Microfilaments, microtubules and endoplasmic reticulum forms endoskeleton of cell. Intracellular exchange. Endoplasmic reticulum forms intracellular conducting system. Transport of materials in cytoplasm from one place to another place may occur through the endoplasmic reticulum. At some places, endoplasmic reticulum is also connected to plasma membrane. So, endoplasmic reticulum can secrete the materials outside the cell. Rough endoplasmic reticulum provides site for the protein synthesis because rough endoplasmic reticulum has ribosomes on its outer surface. Lipid synthesis. Lipids, cholesterol and phospholipids synthesized by the agranular portion of endoplasmic reticulum, which means smooth endoplasmic reticulum helps in the synthesis of lipids like cholesterol and phospholipids. Endoplasmic reticulum also helps in the synthesis of lipoproteins and glycogen. Endoplasmic reticulum also helps in cellular metabolism. The membranes of the reticulum provides an increased surface for metabolic activities within the cytoplasm. Formation of nuclear membrane. Fragmented vesicles of disintegrated nuclear membrane and endoplasmic reticulum elements arranged around the chromosomes to form a new nuclear membrane during cell division. Formation of lysosomes, Golgi bodies, and some microbodies. Endoplasmic reticulum helps in the synthesis of lipoproteins and glycogen, also helps in cellular metabolism. Endoplasmic reticulum also helps in the formation of nuclear membrane and also helps in the formation of lysosomes, Golgi bodies, and some microbodies. Endoplasmic reticulum also helps in detoxification, particularly smooth endoplasmic reticulum concerned with detoxification of drugs, pollutants, and steroids. Cytochrome P450 in endoplasmic reticulum act as enzyme which function in detoxification of drugs and other toxins. Endoplasmic reticulum provides the precursor of secretory material to Golgi bodies. Golgi complex. Golgi complex. The scientist Carmelo Golgi in the year 1898 first observed densely stained reticular structures near the nucleus. These were later named as Golgi bodies. Golgi body also named as lipochondria because these are rich in lipids and also called dictyosomes are plant Golgi bodies. The cytoplasm surrounding Golgi bodies have fewer or no other organelles. It is called Golgi ground substance or zone of exclusion. Carmelo Golgi is the first observer of Golgi bodies. Golgi bodies also called lipochondria because rich in lipids. The plant Golgi bodies are called dictyosomes. The cytoplasm surrounding Golgi bodies have fewer or no other organelles. Structure of Golgi bodies. Golgi complex is made up of three parts. Number one, cisternae. These are flat disc-shaped sac-like structures. Many cisternae are arranged in a stack parallel to each other. The diameter of cisternae is 0.5 micrometers to 1 micrometer. Dense material inside cisternae is called nodes. Various number of cisternae are present in Golgi complex. Golgi cisternae are concentrically arranged near the nucleus. Convex surface of cisternae which is towards the nucleus is called cis phase or forming phase. Concave surface of cisternae which is towards the membrane is called transphase or maturing phase. Golgi complex made up of three parts. In those, very first is cisternae which diameter is 0.5 micrometers to 1 micrometer. Convex surface of cisternae is called cis phase or forming phase. Concave surface of cisternae is called transphase or matured phase. The cis and trans phases of the organelles are entirely different but interconnected. Second one, tubules. These are branched and irregular tube-like structures associated with cisternae. Vesicles. Transition vesicles and mature vesicles are two types of vesicles which are present in Golgi complex. Functions of Golgi complex. Cell secretion. The chief function of Golgi complex is secretion or exportation of macromolecules. Golgi body receives materials from endoplasmic reticulum through its cis phase. These materials are chemically modified by Golgi body. For example, glycosylation or glycosidation of proteins and lipids takes place in Golgi body and it yields glycoproteins and glycolipids. After chemical modifications, materials are packed in vesicles. These vesicles are pinched off from transphase of Golgi body and discharged outside of the cell. All the macromolecules which are to be sent outside the cell move through the Golgi body. So, Golgi body is termed as director of macromolecular traffic in cell or middleman of cell. Golgi bodies and endoplasmic reticulum collectively helps in the formation of lysosomes. The synthesis of cell wall material also done by Golgi complex. Cell plate formation or phragmoplast formation during cell division also by Golgi complex only. Formation of acrosome during spermiogenesis. Formation of male gametes also one of the function of Golgi complex. The Golgi apparatus principally performs the function of packaging materials to be delivered either to the intracellular targets or secreted outside the cell. Materials to be packed in the form of vesicles from the endoplasmic reticulum fuse with the cis phase of the Golgi apparatus and move towards the maturing phase or transphase. A number of proteins synthesized by ribosomes and the endoplasmic reticulum are modified in the cisternae of Golgi apparatus before they are released from its transphase. Golgi apparatus is the most important site of formation of glycoproteins and glycolipids. Lysosomes. Lysosomes. Lysosomes are membrane bound vesicular structures formed by the process of packing in the Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes like hydrolyses, lipases, proteases, carbohydratases, optimally active at the acidic pH at 5. These enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acids. With the exception of mammalian RBC, they were reported in all animal cells. Lysosomes are rich in all types of hydrolyses and those are active at pH 5. In plant cells, large central vacuole function as lysosome. So in higher plants, lysosomes are less frequent, but number of lysosomes is high in fungi. Periplasmic space. The space between cell wall and cell membrane in bacteria may play similar role. Lysosomes are spherical bag-like structures which is covered by single unit membrane. They are large sized in phagocytes, especially WBC, white blood cells. Lysosomes are filled with 50 different types of digestive enzymes termed as acid hydrolyses for digestion of all types of macromolecules. Lysosomes are highly polymorphic cell organelles because lysosomes have different physiological states. Types of lysosomes. The very first one, primary lysosomes are storage granules. These lysosomes stores enzyme acid hydrolyses in the inactive form. Enzymes synthesized on ribosomes in cytoplasm. These are newly formed lysosomes. Digestive vacuoles are heterophagosomes. These lysosomes formed by the fusion of primary lysosomes and phagosomes. These are secondary lysosomes. Lysosome with inactive enzymes are called primary lysosome. They stores food. Secondary lysosomes are fused form of primary lysosomes and phagosomes. Residual bodies. Lysosomes containing undigested material are called residual bodies. These may be eliminated by exocytosis. Autophagic lysosomes are autophagosomes. Lysosomes containing cell organelles to be digested are known as autophagosomes. Functions of lysosomes. Lysosomes helps in intracellular digestion, heterophagy. This is a digestion of foreign materials received in cell by phagocytosis and pinocytosis. Autophagy, digestion of old or dead cell organelles. Autophagy also takes place during starvation of cell. Extracellular digestion. Lysosome of osteoclast, bone eating cells, dissolve unwanted part of bones. Extracellular digestion also occurs by fungal lysosomes. Cellular digestion or autolysis. Sometimes all lysosomes of cell burst to dissolve the cell completely, so lysosomes called as suicidal bags of cell. Old cells are removed by autolysis. Unwanted organs of embryo are destroyed by autolysis. Lysosome digests the tail of tadpole of frog during metamorphosis. Vacuoles. Vacuoles. Vacuole is the membrane bound space found in cytoplasm. It contains water, sap, excretory product and other materials not useful for the cell. The vacuole is bound by a single membrane called tonoplast. In plant cells, the vacuoles can occupy up to 90% of the volume of the cell. In plants, the tonoplast facilitates the transport of a number of ions and other materials against concentration gradient into the vacuole. Hence, their concentration is significantly higher in the vacuole than in cytoplasm. Vacuole is membrane bound space found in cytoplasm which stores excretory wastages in the cell. Vacuole covered by a single membrane called tonoplast. In amoeba, the contractile vacuole is important for excretion. In many cells, as in protists, food vacuoles are formed by engulfing the food particles. Key points on endomembrane system. Endoplasmic reticulum, Golgi body, lysosomes and vacuoles are considered together as endomembrane system because their functions are coordinated. The vacuoles store and concentrate waste products. The same are segregated from the living part of the plant cell. RER, rough endoplasmic reticulum, frequently observed in the cells actively involved in protein synthesis and secretion, whereas smooth endoplasmic reticulum is observed in cells actively involved in lipid and steroidal hormone synthesis. Golgi body receive the materials from endoplasmic reticulum through its cis phase, and these materials are modified by Golgi body for formation of glycolipid and glycoproteins, which is called glycosylation. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes like lipases, proteases, carbohydratases, optimally active at the acidic pH. In plant cell, the vacuoles can occupy up to 90% of the volume of cell. It contains water, sap, excretory products and other materials not useful for the cell. Up to here, cytoplasm and endomembrane system like endoplasmic reticulum, Golgi complex, lysosomes and vacuoles are completed. In next video, I will explain you about mitochondria and chloroplast. Thank you very much.